Hello and good evening, and welcome to today's event from the Jesus College Intellectual Forum as part of this year's Cambridge Festival. My name's Julian Huppert. I'm the director of the Intellectual Forum here at Jesus, which you can see behind me on a somewhat sunnier day and with a bit less snow around. We are recording this event for people who can't make it with us right now. Now, Jesus College is an ancient institution. It's seen a lot of changes in lifestyles, in technology. We were a 12th century nunnery and became part of the university as a college in 1496. So we've seen civil wars, world wars, pandemics, huge changes in every possible way. The Intellectual Forum is rather more recent. We were set up 6 years ago to get people to think and talk about important issues and to reach out beyond the boundaries of our college. We've brought in some utterly amazing speakers from around the world, everyone from fashion designer Jimmy Choo or Helen Clark, who ran New Zealand and then the United Nations Development Programme. We've had Belinda Wilkes, who ran NASA's Deep Space X-ray Observatory, and the Assistant Director General of the World Health Organization, Peter Simpson, who talked about vaccine equity around the world, obviously a crucial time. And there's been many, many other things. We also show off some of the amazing work that's being done by our own academics, whether that's using synthetic biology to come up with a new, safe, clean way of dyeing clothes, to how we can actually use the financial systems to save us from climate change and other risks that we face. There's a lot more, and if you want a real treat, have a look at the Jesus College YouTube channel, where almost all of our events are captured and where this will be available in a few days' time. For this Cambridge Festival, we're doing a total of 6 events on very different fields, showcasing some of the amazing people from the Intellectual Forum and the wider college. And they cover everything from improving childhood nutrition to making medieval medical charms via sustainable finance, quasi-crystal games, and digital productivity. So there's something there for everyone, and we hope you'll join us for many of them over the coming days. And today I'm really delighted to welcome Dr. Kim Liu. So Kim's been here at Jesus since 2011, originally as a student, and now as one of our Star College postdoctoral associates. His day job is at the MRC Laboratory of Molecular Biology, where he works on synthetic biology. How do we use biological methods to do new things? So he's going to talk for a while about his work and how and why we can break the normal rules of protein biosynthesis, and then take questions. So please put your questions in the Q&A section on Zoom, and we'll come to those later. So Kim, it's really wonderful to have you with us. Over to you.
Hi, can you all hear me and see me? I think I'm here now.
Yes.
Excellent. All right. Hi, everyone. I'm delighted to be here tonight to talk to you about this concept in synthetic biology, which I've recently turned my research interest towards. And this concept has been knocking around for about 20 years or so in quite a simplistic manner, but I'm going to talk about it. I'll talk about a relatively recent development, which has really pushed this technology to the furthest it's ever been. And tonight, I understand there's a large variety of folks from different backgrounds tuning in tonight. So I'm going to start from the basics, and I'll explain what these rules are before describing what happens when we try to break them. And hopefully by the end, you'll appreciate the potential that this technology might have in the future for healthcare and even potentially producing new materials. For myself, I've enjoyed drawing this graphic that you can see on the screen. And if by the end the graphic makes sense to you, I'll be pretty happy, because I can imagine that it might be a little bit complicated at this stage in the game. So as Julian said, I'd be really happy if you wrote questions down in the chat, not in the chat, in the Q&A box as we go, and I'm excited to hear about them at the end. But with that, let's begin with life itself. I really enjoyed making this slide and sort of browsing through all of these beautiful photos to pick out a couple that I really liked. But essentially, what I want this slide to do is just help you think about why everybody, enjoys watching David Attenborough's shows, frankly. Think about the vast diversity of life that you can see in these pictures, that you've experienced, that you know about, your favorite animals, but also to microorganisms, fungi, and things that you can't see with your eyes. Think also about the range of abilities that life performs, everything from breathing underwater to flight, and then also consciousness in humans, maybe other animals as well. All right. I'm going to sort of, in the sort of tree of life, give a special mention to bacteria, just because they're particularly important in today's story and arguably, the most dominant life form on the planet. And also, of course, to viruses, since COVID has affected our life so profoundly in the last 2 years. But these things are all part of this magnificent story, so to speak. Also, I would have you think about complexity. Here, I've just got a network sort of infographic of all the processes that go on in a single compartment of a human cell. Right? And the compartment that I've got here is the mitochondria, which some of you may know as sort of the powerhouse of the cell. I don't know about you, but every time I see things like this, I'm always sort of mildly mind blown by how complicated a cell can be. And this is only one compartment of a cell. Right? There are other compartments in the cell, and there are all sorts of other molecules floating around as well. Right? So just for interest, I've got here, just this little bit here in blue, describes what happens when sugar is burnt in the mitochondria and converted into energy. Right? And around here, for example, there's some fats being burnt as well in a similar way. Right? But the point of all this, of the slide before, and of this network showing all of this complexity, is just to hammer home this fact that all living things and the processes behind them, all of these things are based on proteins. Every single step you see in the networks is carried out by a dedicated protein. Right? Proteins form the structures of all living things, hairs, horns, feathers. They organize the other chemicals like fats, sugars, any other things that the cell needs to survive are all processed by proteins. So clearly they're incredibly important, and they are the star players of our story today. So first we need to know how to make them. And so I really like Lego as an analogy for how proteins are built. Right? Because there are chains of different building blocks, which we call amino acids, and they're chemically linked together, just as Lego bricks are all different colors, but they stick together in the same way. They have the same general shape, but they connect to each other with the same chemical link, right? Just like Lego. And just like Lego, they have to go in a certain direction. You can't put Lego the wrong way around on each other, right? And for the analogy, the different colors represent different building blocks. In reality, these are represented by different kinds of chemical species attached to the amino acid. So I've just got four things connecting together, but a normal protein is going to be hundreds of these amino acids long, hundreds of building blocks chained together. All right. And as this gets longer and longer, it's the order of these proteins, the order of the colors in this protein, or the order of these amino acids, which makes the protein unique, right? A certain order of colors will allow you to cut up a sugar molecule so that it goes into the next step, which will lead to energy. A different order of colors leads to, producing a protein called keratin, for example, which is what is found in human hair. Okay. And then where the Lego analogy breaks down just a little bit is the fact that that little tower of Lego blocks that I have shown before, that's not going to ever bend. But in reality, proteins can also bend, right? They start to form pretty impressive sometimes 3D shapes. And these 3D shapes are really important because the 3D shape is usually what permits a protein to act in the way that it acts and give it the function or the ability that it requires to carry out the process that it's trying to look after or modulate. Okay, and I'm just going to give a quick shout out to AlphaFold by Google DeepMind at this point. And if that's something you haven't heard of, it's a very powerful computer algorithm which actually is able to predict the 3D shape of proteins from just the sequence of amino acids. And this is a real milestone in biology. It makes understanding the function of proteins much more important, and this is crucial for understanding disease and health. Right, so this is what a protein is, And this is how they tend to act, and they are very important in all of the processes that we see in life. Well, we also need instructions, right? Lego always needs instructions, and instructions are of course carried by this wonderful molecule DNA. Right? It carries the instructions that give the order of the building blocks which constitute our proteins, and this is what we call the genetic code or genetic material. Okay, it contains only 4 letters: A, C, G, and T. And why this was such an important structure when it was first discovered is the structure allows you to understand how life can copy this genetic information and pass it down through the generations. I really like this. This is a little excerpt from a letter that Francis Crick wrote to his son at the time. And he said, in other words, we think we have found the basic copying mechanism by which life comes from life. Right? And it can do this because it does this thing called base pairing, where these letters are only allowed to be paired in 2 ways, G with C and A with T. So the double-stranded nature, the double-stranded helix of DNA effectively allows a structure that looks like this. But actually for proteins, this doesn't matter as much, but it's important to just recognize that DNA is the thing which carries the genetic code that we're going to be playing around with later on. So the genetic code is exactly like a language. In a language, combinations of letters mean different words. In the genetic code, the 4 letters combine together into 3-letter words only. Okay, so A, C, G, and T can be arranged in groups of 3, and those groups of 3 represent the building blocks or amino acids. So for example, GGG is going to represent the white building block, okay, the white Lego piece, and CCG is going to represent the green Lego piece. All right, and so DNA carries all of this information, and then when the cell reads this information, it will put the proteins together according to the instructions that the cell will then be seeing in the DNA. All right. I've also got TAG here, which represents stop, because it's also important to tell the protein when to stop being made. And actually the stop codon, the stop word will in fact be important to us later on. So that takes us to the sort of first half of this picture. This is an ordinary bacteria reading the genetic instructions, an ordinary set of genetic instructions. And effectively what I've got here is, this is the sequence of DNA, ATG, TCG, TCA, etc. And it reads the genetic instructions, which is the genetic code, and assembles the proteins or the Lego blocks together in the order determined by the DNA. And this is just an ordinary bacteria doing this. Okay, and so the rules that we're going to hopefully think about breaking later on are basically as follows. And in fact, it's actually quite interesting to ask yourselves, if you don't know the answer, how many building blocks do you think there are that exist in the natural world? All right, and the answer is there are only 20. And these 20 amino acids, these 20 building blocks, are encoded for by the same universal triplet genetic code, triplet just meaning groups of 3. Right? And this code, this universal code, gives you all of these things that I showed you earlier in the presentation. Right? And that for me is sometimes, yeah, this is really mind-blowing to me actually, that all of these things come from only 20 different amino acids. And okay, sure, there are hundreds of potential, a protein is hundreds of building blocks long. So if you have 20 options for hundreds of positions, then yes, you can imagine that there are a lot of different proteins. And so far, we know of 100 million different known proteins. And certainly that's the number that's stored in a database at the moment, at this point in time. And certainly there are many being discovered every single day. Okay. If we zoom in to the genetic instructions, this is the whole set in full. And this is the set of instructions that both the bacteria and humans and trees, blue whales, etc., they all follow the same code, right? If you have groups of 3, 4 letters in groups of 3, we get 64 possible combinations, okay? And then you hopefully can appreciate that only 20 of them are needed. So there's going to be some repetition in this code. There are lots of codes, which encode the same amino acid. I should just say the amino acids now, I've gone away from the Lego blocks. They're now just the letters in bold. Okay, so for example, the amino acid leucine, encoded LEU in the left column, is encoded by 6 different codes. So there are 6 opportunities for DNA to tell the cell to use leucine. And there are 6 opportunities also to tell the cell to use SER, which is serine, but there are only 2, for example, for histidine, which is HIS. Okay, and there are some quirks behind why certain amino acids have different numbers of codes, but the important thing is there are more codes than there are building blocks. Okay, so with these rules, just using the rules that we've got, we can already start doing some amazing things. All right, the first thing is you can transfer proteins or functions between different species, between different life forms, just by copying the DNA instructions. So here, this is a picture of a jellyfish, which actually produces the green fluorescent protein that I showed earlier. And it just fluoresces green. So it has this really nice color when it's put under ultraviolet light. And all you need to do is extract this DNA. And I'm not going to go into the details of how you manipulate DNA from a chemical point of view, but let's take for granted that we can just copy the DNA and transfer it. If we then transfer it into a variety of different animals, we can give those animals the ability to basically glow in the dark, right? And this is, there are lots and lots of steps and lots of complexities to think about, but essentially it's the same sequence of letters at the end of the day being read by a different organism using the same genetic language, the same genetic code. Right. This is in some ways, so this is useful if you're trying to track proteins inside human cells in a dish. This is what you can see here, for example, on the top right. But for the other things, turning rabbits glow in the dark perhaps is a little bit of a trivial example, but a really important example is the production of human insulin. So human insulin, as hopefully a lot of you will know, is a treatment for high blood sugar in diabetic patients. Okay, so it's a very, very important drug and is very important for diabetics to manage their illness. And so what we can do is we can take the gene for insulin, which is found in humans, and we can put it inside bacteria. And what that allows us to do is we can get then the bacteria to synthesize lots and lots of the insulin. Okay, and generally biosynthesis is easier than chemical synthesis because the bacteria can cheaply produce the insulin relatively compared to chemists, because all you need to do is provide it with some sugar, some nutrients, and grow it at 37 degrees. To produce the protein or the hormone insulin in a chemical way, in a chemical lab, mixing together chemical reagents, is much more expensive and much more time consuming. I'll talk a little bit more about this problem in a few minutes. But the point here is biosynthesis often is a good way to produce things, because growing bacteria, growing yeast, growing algae, for example, all of these things don't require all that much energy compared to a large chemical plant, which has to process sometimes quite harmful chemicals in order to produce the things that we need. Still playing within the realms of the current rules is CRISPR, which is something that sort of exploded in amongst the general public, I think, recently, and they're known as these DNA molecular scissors or molecular DNA scissors. All right, it's a mechanism by which you can target specific DNA sequences, cut them, and then reintroduce new pieces, a bit like a patchwork device. And it's incredibly efficient. The reason it's received so much praise and awarded so much prizes, and including the Nobel Prize, is because it's incredibly efficient compared to methods that came before it. So potentially the really, really important thing that this sort of unlocks the possibility to do is make small changes to DNA, which allows curing single mutation genetic disease. So there are a bunch of genetic diseases in which the proteins which are important have one single error, which leads to the wrong building block being incorporated. So CRISPR is a relatively straightforward way, at least so far in cells and in animal models, to change that amino acid back to the correct amino acid just by changing the DNA sequence. Okay? And this is especially good when only a single building block is wrong. If you have to change multiple building blocks, this becomes a little bit more complicated, right? But these are companies, these logos here are companies that are currently putting these therapeutics through clinical trials as we speak. But as well as just healthcare, CRISPR also allows other more imaginative and sort of out of the box problems that can be solved as well. So one of these things, for example, is organ transplantation. And this company, eGenesis, spends time using CRISPR editing pig organs so that they're more amenable to transplantation into humans. Right? It doesn't sound particularly appealing, but there is a big shortage of organs in general, and transplantation shortages is a problem that requires solving. And one of these solutions might be to make a pig's liver, for example, a lot more human friendly. Right? This other one is producing fuel from algae. And I really like these three on the side, which are effectively editing plants so that they are more effective as food sources, as nutrition for humans. So one which produces healthier oils, one which genuinely is editing a mustard green to make it more tasty, and also this one which customizes seeds so that they can grow in any conditions. So they can grow in drier conditions or wetter conditions, that kind of idea. So the point of this slide is just to show that even within the current rules of protein synthesis, there's a lot of untapped potential, and there's a lot of research into developing proteins and changing them so that they can achieve new things which benefit humans in general. Okay. So this is what I'm going to show here. On the left is the same bacteria that we had earlier, and I just want to indicate that even within those rules, we have CRISPR, we have all sorts of genetic modification. Amongst the 20 currently existing building blocks, there's a lot of untapped potential. There's a lot of things that can be done and are being done to try and develop new technologies for healthcare and for energy supply, for nutrition, etc. But what I want to talk about is potentially breaking rules and seeing if what we can do is expand the genetic code. All right. So in this case, what I've got on the right is, instead of producing the sort of standard Lego brick that I've got on the left, I want to include some fancy Lego bricks, right? One which is sort of wide and thin as opposed to just the sort of normal shape, and one which is slightly elongated, which has a longer backbone effectively, right? So I'm asking the question here, given that we have all of this potential from 20 amino acids, what happens if we add 10? What happens if we add another 20? Right? If you think back to the table that I had earlier, remember I said that there are 64 possible letter codes, and only 20 of them are currently being used for amino acids. Potentially, there's up to 44 new amino acids that we could potentially put into this code. And that's a whole bunch of new chemistry, a whole bunch of new structures potentially, which we haven't yet been able to discover. And crucially, nature would never be able to discover at this stage in time. Nature only has those 20. And as far as we can tell, there's no hint. Well, there are some hints that they might be changing them slightly, but generally it's going to be much more, it will be many, many more millions of years before it starts to incorporate more amino acids, I think. So how can we do this? And so I said there was a simple way of doing this, which has been sort of floating around for a while. And that's to hijack the code that says stop. Okay, so what we do is we choose a specific position on the protein which we want to put in the new amino acid. We change the code at that position to TAG, because TAG usually encodes stop. So some of the time, instead of saying stop, you could then get it to insert a new unnatural amino acid. Okay, you also need to add a gene which allows the amino acid to be carried. That's also an important part of it. But effectively, what I've got here is a sequence with TAG in the middle, and usually that would just say stop. But in this case, we can get in this single disc into this protein. And already with this technology, we can do quite a lot. So for example, if that disc was fluorescent or sort of can be lit up inside a cell, that protein could then be tracked inside a cell. There are no fluorescent amino acids, so this is not possible with the current existing amino acids. But if this amino acid, the disc that I've got here, if the orange disc were able to light up, we could then follow that protein around inside a cell, which would be really interesting to see if the location of that protein was important for what it was doing, for example. Okay. Another more complicated example is identifying the targets of enzymes. Okay, so enzymes are proteins which perform chemical functions, and the targets of these enzymes, so the enzyme is this green blob, I should say. The targets of these enzymes are often some specific chemicals floating around the cell, which the blob can sometimes go and cut or process or add something to. Okay, so here I've got an enzyme called a hydrolase, and that's particularly an enzyme which cuts up other molecules. And it just so happens that 14% of these hydrolases are active drug targets. So this is a good place to look for new therapeutics and new ways to cure disease if we can understand where these enzymes are currently cutting. So if these enzymes happen to cut substances or chemicals or proteins even that are involved in a disease, it would be really useful to know that. Okay, so here we have, for example, this green enzyme is going to come along and cut this little chain at the red position. Okay, and it might be that that chain is problematic for some disease. So it'd be really useful to know if this was something that was happening inside a cell or could happen inside a cell. Okay, so if we use an unnatural amino acid instead, we can replace the cutting part of the enzyme with a light sensitive trap. Okay, so we could replace it with a trap and then put a cage around it, which is light sensitive. So we activate the trap by shining light on the cell. So once the cell sees light, that trap will uncage itself and then the trap will become active. Okay, so that's what I'm just showing by the circle disappearing off the star. And then instead of cutting this, this molecule, the star might instead stick to the molecule and capture it. And then if we capture the protein and the enzyme in question, we would then be able to identify through a range of chemical means what the target of that enzyme actually was. And so that's really, really useful for trying to understand whether or not this enzyme could be a new drug target in the future. Another really cool example is towards enzymes or proteins that perform new reactions. And these are reactions that are invented by humans, which we don't see in nature. And don't get me wrong, nature does amazing chemical reactions, lots of reactions that humans find very, very difficult. But there are some reactions that humans have invented which we don't see at all in nature. And one of these is this reaction called photoredox catalysis. Okay, and this is a very, very important chemical process. It's allowed the transformation of all sorts of different chemicals. Lots of the reactions that I tell my students don't happen and are not allowed are apparently just possible with this photoredox catalyst. So it really does change a lot of sort of fundamental chemistry and how you can build new molecules. Anyway, you can see that this catalyst looks like has these sort of double hexagons attached to a central metal core. In this case, it's iridium. And these hexagons don't appear in this configuration in the amino acids. So if we want to imitate this kind of catalyst in a protein, we need an unnatural amino acid. And you can see that here in this protein, where we've got these double ringed structures surrounding this metal atom. And so this protein, by using this unnatural amino acid, is able to imitate the reaction site of this photoredox catalyst. And maybe one day we could evolve this protein so that it does some more interesting chemistry. Cool. But these are relatively simple things. There's a lot that can be done, but relatively these are simple ideas compared to the ultimate goal of expanding the genetic code and breaking it so that we can do encoded chemical synthesis. Fundamentally, if you think about protein synthesis, it's an incredibly efficient and accurate and reliable method of making long chemical chains. Fundamentally, that's what proteins are, just long chains of different chemicals, right? And inside a cell, they're very, very rapidly, very, very reliably made. If they're made wrongly, the cell will really suffer, described by DNA, which is the set of instructions inside the cell, right? And so just to compare human chemists to the cell, in this regard, in the specific regard of making proteins, you can see the cells outcompete humans on almost every level, right? The maximum length is longer. It's way faster. It doesn't need as high temperatures. It also uses sort of reagents or materials which are by definition non-toxic and biodegradable because they basically exist inside living things. Whereas a human chemist has to use organic solvents which are harmful to both humans and also to the environment. All right. And so the question then becomes, why stop at amino acids and protein chemical bonds? And this is what I mean when I talk about, or I have talked about this idea where we can access new chemicals or new polymers which currently can't be synthesized, or at least try and synthesize polymers which we don't, which we know are very useful, but are quite expensive and difficult to synthesize. For example, Kevlar, which is a really, really impressive polymer, which is used in bulletproof vests or strengthening steel, for example. It's a very, very strong polymer, but it's not something you see every day because it's quite expensive to make. In principle, if you could hijack the genetic code, rewrite the rules so that you could incorporate the building blocks that make Kevlar, you might be able to, for example, get a bacteria to produce something like Kevlar, which would potentially be much easier, much cheaper, and much healthier for, more sustainable for the world in general. Okay, but if we want to do the encoded chemical synthesis, we need to do more than one building block, because the stop codon method only really works well for one building block. Stop is a special case because there's nothing to replace or interfere with the codon other than stop. But if you want to use the other codes, we're going to run into this problem, which I've drawn on this slide. Okay, so I'm just going to talk through it. If you want the unnatural protein using these TCA and GCG, so the bacteria in the corner wants to encode the purple Lego block with TCA and the flat orange block with GCG. If you put in the DNA that says ATG, TCG, TCA, et cetera, some of the time you're going to get the protein that you want, and that's fine. A lot of the time, you're going to just get the actual amino acid that nature normally intended. Right? So you're going to get this interference where you don't have as much of the protein that you want being produced. More importantly, if you have TCA or GCG inside the important protein sequences that the cell has already, those essential proteins are potentially going to be damaged by putting in the wrong amino acid. Remember, the sequence of amino acids is really important for how the protein works. If you change those sequences, that protein may stop working. And even if you change only a few of them, sometimes this can cause the cell to die. So if you try this for other codes which are already used for other amino acids, you generally tend to just kill the cell. So this is a really, really big challenge in trying to get encoded polymers or trying to get multiple amino acids and unnatural building blocks at the same time. So how do we solve this problem? Okay, we have to remake the genetic code. All right, and so how we did this is we looked at this, this example, which is serine. And serine, you can see SER, has 6 codes available to it. So we thought, okay, we'll take away 2 of those codes. We'll take away TCA and TCG. And we'll also take away TAG because that's the one we've already been using. Okay, so what we're going to do is we're going to take them out of all of the positions that you would ordinarily find them in the bacterial genome. Right, the genome is the full list of instructions for all the things inside the bacteria. Okay, and that's 4 million letters long. So the number of edits you have to make comes out to 18,214. And that's just too much to do with any gene editing technology that currently exists. Even Crispr wouldn't be able to do that very easily at all. Okay, so what the group decided to do is make all 4 million letters again from scratch. So keep most of it the same, but just change every time you see TCA into an AGT, so the serine, or TCG into AGC, and TAG into TAA. So every single time you saw these codes, which I'm crossing out, we changed them and put in an equivalent code, which would give you the same building block inside the cell. Okay, and so it was determined that actually the way to do this would just be to make all 4 million letters from scratch. And so our group entered became interested in synthesizing whole genomes. This is a particularly complicated slide, so I'm not going to dwell too much on it. But the idea behind the process is you just take 10,000 letter long pieces of DNA. So that's about as long as you can buy at this stage in the game. Right, so you can buy pieces which are about 10,000 letters long. And effectively what this method is stitching all of those 10,000 pieces together into sequentially bigger and bigger pieces until eventually you replace the whole genome. Okay, just to be clear, the first step is sort of a standard step that's done very widely. Also, this final step is not particularly difficult. The difficulty was this step here, which was sort of, this is the sort of the stage at DNA synthesis, if you like, which was particularly challenging. And sort of the amusing thing you can take away is that they decided to call it Genesis at this point. So we've made a whole genome, which is completely synthetic. This bacteria contains DNA that all originated outside of the cell. It was made by humans first and then inserted step by step into the bacteria. Okay, and this Sin61, so named because it only has 61 of the original 64 coding letters, can produce proteins with unnatural amino acids using TCA, TCG, and TAG. Before this was not possible, those cells would have just died. You can also produce proteins with up to 3 different amino acids in one protein, and you can actually also chain up 6 unnatural amino acids together in a row. That doesn't sound like very much, but there are some other complications which what this single solution doesn't address. I will talk a little bit about that later. Okay, but this is Sin61 then on the right here, who is now reading an expanded set of genetic instructions where we've deleted some of the codons from its genome so that it can happily make TCA, TCG, and TAG unnatural amino acids. And these are the first steps to getting to this stage where we can just start chaining together different chemicals to make chains, new chains of chemicals. Okay. There was one also interesting side product or side effect of doing this recoding, okay, which is that it resists virus infection. So how do viruses work? Viruses inject their DNA into their host cell. COVID would have done exactly the same, but here we're looking at bacteria for human cells. Here we're looking at bacteria viruses into bacteria. They inject the DNA and the cell is forced to make bacterial proteins. Those bacterial proteins then sort of form the virus inside the cell and then explode out of the cell and then go on to reinfect other viruses, other cells. Okay, and that's the same for COVID as it is for these bacteriophage, which are viruses which infect bacteria. Okay, so if you think about how Sin61 works, though, the bacteriophages, the virus's genetic instructions contain these 2 codons, TCG and TCA. Right? There's no reason, because it uses the same genetic code that we talked about before as all of the other domains of life. There's no reason for it to not use TCG and TCA. But Sin61 doesn't know how to read TCG and TCA anymore unless you provide a new unnatural amino acid. And that means Sin61 will never make any of the virus proteins correctly. It will accidentally make them all wrong, and then the virus is unable to duplicate itself and unable to infect other things. And this is what you can see in this picture here. When bacteria is healthy, it's nice and cloudy. Okay, so when there's no virus added, you can see that it's nice and cloudy. And this is a normal bacteria on the left. On the right, you see that after you add the virus, all the bacteria dies and you can't see it anymore. Okay, or it forms a very sort of small pellet at the bottom and it doesn't float. On the right, you have Sin61. And even if you add the virus, it still grows fine. And that's what you can see also in this graph. On this y-axis, you see live virus, and you can count the live virus and over time after infection, right? Initially, you always see the virus amount go down because the viruses are going inside cells ready to make more virus. But only in normal bacteria, the red line, do you see that the bacteria eject more virus and therefore more virus is being produced and the infection continues. Whereas in Sin61, you see that all these viruses are just constantly absorbed, but they're never able to make new viruses. And so that was a really, really cool sort of side effect of this process that we were doing. Okay. So quickly before I finish, I'll just briefly talk about what needs to happen next, because as I sort of alluded to earlier, there are still some things which are holding us back. And here we need to look closer at the mechanism behind the protein manufacturing factory in the cell. Okay, so this picture is a little bit complicated, so just bear with me. The amino acids are now circles and stars instead of Lego blocks. And these are enzymes or proteins, these AARSs, which pick up the amino acid and put them onto these cross things called tRNAs. Okay, so that's what's necessary when every time the genetic code is being read. I never really talked about any of this mechanism, but this is what's going on. This protein in black, the sort of the square things, pick up the circles, put them onto the crosses, and then the crosses read the genetic code. The ribosome then stitches all of the protein, the amino acids together to get this protein. Okay, and so you would also want for the unnatural amino acids, which are the stars in this case. Okay, but a crucial problem here is we need everything to be orthogonal, and that's what the O stands for. And what I mean by orthogonal is these things can't overlap with each other. Okay, if you think about the original code, if the amino acids could be loaded onto the wrong tRNA by a certain enzyme, then you'd have trouble. You'd have basically unreliable reading of the genetic code. And so it turns out it's quite a difficult thing to keep all of these different enzymes orthogonal to each other. That is to say, they only act on a single amino acid. If they act on different amino acids, it means that your genetic code kind of can't be read accurately anymore. Okay. And also the ribosome has a preference for certain types of chemical bonds. Okay, so the reason we couldn't go now and try making Kevlar is because the ribosome wouldn't be happy putting the bonds together. Okay, and the solution to these problems is we need to just evolve these ribosomes and evolve these proteins until they start to accept, and we have methods of doing this. This is the current research that we're getting up to at the moment, accepting more unnatural amino acids. Okay, and this is why we could only achieve 6 in the earlier example, and these are the next hurdles that need to be overcome in order to be able to produce the long encoded non-canonical biopolymer, or the long chain of just chemical things stitched together. Okay, and that's where I'm going to finish and leave a summary. If you take away just one thing from today, take away the fact that all of life uses only 20 building blocks to build all of its proteins, all of its functionality, and it uses the exact same genetic code. Okay. If we break the rules and rewrite them, we potentially can get abilities for these proteins, functions for these proteins beyond which nature can currently evolve. Nature only plays with 20 amino acids. What can we do with 10 more, 20 more, maybe even 50 more? Okay. And there's this dream that we can produce any kind of chemical, maybe a drug, maybe a new kind of plastic, which is biodegradable, and we can get living things to just build these molecules for us instead of having to use chemical plants and high temperatures, high pressures, etc., to produce these things. Okay? And Sin61 is the first step towards making these unnatural polymers, and it just happens also to be resistant to viral infection. And with that, I'm going to close, and I'm really looking forward to seeing some of the questions, hopefully, that are popping up. And thank you very much for your attention. This has been really, really great.
Thank you very much, Kim. I mean, that's been really fascinating and really interesting to hear both how the natural systems work and some of the vision for what you're coming to. We do have lots and lots of questions. I'm guessing from some of the questions that people who know about this field and others that don't. I might try and go in slightly chronological order. So firstly, you talked about the fluorescent protein, green fluorescent protein exciting. Can you transfer that into humans?
Okay, so the first thing you need to do is you need to copy that DNA using a process called PCR. Okay, that's probably the easiest way to do it, is to copy all of the DNA using a bunch of enzymes which basically can run along and copy the DNA inside the inside the jellyfish. You would then cut that piece of DNA and try and insert it into the genome of the recipient cell. Okay, so for bacteria, that's not so difficult. You can just put it inside a piece of DNA, and usually that piece of DNA can just go straight into the bacteria without too much trouble. For a multicellular organism, this is a lot more complicated. You need to inject it into embryonic stem cells so that the multicellular animal then develops and keeps that gene and then keeps producing it in all of the cells in its body. Right? So it's a very, very long process. You also need to think about what are called promoters, which are sequences of DNA which control the expression or the production of these proteins. So there are lots of things you need to get just right for it to work, but it is something that can be done at this stage.
But
I hope that's okay. It's quite brief, I think, as an answer, but yeah, in the interest of time.
But it's not a way that I'm going to get my hand to grow fluorescently for glow in the dark.
So right, so you can't use this method to edit a living person right now. You have to edit the stem cells before the embryo is developing. So we can't now make at the moment our hands grow green for them at the moment.
There we go. What a shame. Now, there's been a number of questions about tag and stop genes. So there's one question about just why does tag mean stop? And I'd like to bring that with a question from Bowen Huang about how do you get TAG to be read as unnatural amino acid instead of stop? And then just to throw in a third question from Daniel Askew, if it does that, how does it now stop?
Right. So, okay. The first question, which is why does tag mean stop? I don't think there's an answer to the question why. It just does. It just happens to be the sequence, which means stop a lot of the time. Okay. I didn't mention this. Actually, it's a really good question from thanks, Dan, which is that there are in fact 3 stop codons available, and I didn't point that out when I was presenting, and I should have. There are in fact 3 different stop codons. So in principle, we could use any of the other 2 to continue stopping. In the case where you're adding the unnatural amino acid to a system which is expecting to stop, you can run into trouble. That is a problem with the system, is that occasionally, instead of stopping, the protein will accidentally put in the unnatural amino acid, and that's a problem for the cell. And then the final one, yeah, how do you get the tag to be read as the unnatural amino acid? So that's the second to last slide with all of the different stages with the crosses and the circles. So you need to, when I said in the slide, you need to include a gene for the carrier of the amino acid. And that's kind of what that's the process that you need to go through. You need to produce an amino acyl synthetase, the enzyme which loads the unnatural amino acid and load it onto a tRNA, which recognizes the TAG code in the genetic code. And that's a complicated process, but that is also doable and has been done for some number of years right now.
Fantastic. So that's
that makes sense.
From Malachi Jones. Does putting your disc into the protein chain change how it folds significantly? You know, can the enzyme still do the same cutting if the folding is different? Yeah,
that's a fantastic question. And the answer is you don't know until you try, but you can make some educated guesses about what you might expect. So for example, if you put the disc far away from the business end of the protein, you would probably expect the business end to still retain its shape, and then maybe a small perturbation around the back end of the protein, for example. So that would be good if you wanted the light. But the example you gave is, how do you know that that cutter is still going to be the same shape? And the answer is, you're right, it might not be exactly the same shape. But it just so happened in the example that I showed, which was recently published, it did work out okay. And it did work out that the shape of the enzyme didn't change too much. But that's not something you can take for granted. You're absolutely right.
Right, so there's a lot of detailed engineering in the folding, and you showed some of the amazing work with DeepMind, so yeah, might help a bit.
Yeah, that would indeed be very helpful for that, yeah.
So there's then a couple of questions which I think, I mean, my own work was on DNA. All this protein stuff is far too complicated for me, and I think they're related questions. One is, why only replace 3 of the codes with new ones instead of 44, which I think is equivalent to another question about, if you're creating all the DNA from scratch anyway, could you compress all the way down to SIN21?
So this is something I didn't mention too much, but it just so I didn't mention how sick SIN61 was when we first made it. Okay, and so this is one of these sort of this is a big problem with sort of complexity in biology. In principle, yes, you can compress all the way down to SIN21, but we only wanted to try compressing down to 61, and even when we compressed down to 61. We ended up with a bacteria that grew about 2 times slower, right? So the bacteria was sicker just from changing these sequences. In principle, the proteins were being made were exactly the same, but something was happening to the SIN61, which made it grow more slowly. And so the expectation is, if you did go all the way down to SIN21, you probably would end up killing the cell, because there's some complexity, I think, in the way the different coding sequences interact with each other, which we don't yet know too much about. Does that make sense? It's a thing that we're really unsure about. The way we get Syn61 to be useful, by the way, is to do some artificial evolution. So we've evolved it, so now it grows quite healthily. But the problem with the evolution is we have no idea what the changes in the genetic code actually did. So they've changed all sorts of different genes, but we don't really know what any of those genes are related to in terms of what's happened to this bacteria. So there are some strange things going on which we still don't understand. And that's why trying to go straight for Syn21 would be quite difficult. I should add that each time you want to try making these genomes, it will cost you about £1 million. So it's also very, very expensive from that point of view. So.
Certainly from my own research experience, Kim and I did our PhDs with the same person at slightly different times. There's a lot in biology which it turns out we just don't really know or understand yet. I still remember when junk DNA meant DNA which has no purpose whatsoever. And I think we now define junk as we just probably don't understand it, and some of it's important and some may not be. So I think there's a number of challenges there.
Yeah.
There's loads more questions flooding in. I'll try to get through as many of them as I can. Ben Lewis says, thanks for a great talk, Kim. What amino acids and for what purpose are you most excited to add to the Syn61 bacteria?
Good question. I think the honest answer is any that work, I guess. But I think for now, and this is quite a technical thing, is we're quite excited to try and get what's called the beta amino acid. And what that is effectively an amino acid which has been stretched out a little bit. It has an extra carbon atom, and that just means you can make the backbone slightly different in the protein. And the reason it's been quite a difficult thing, it's just historically been a very, very difficult thing to do. And lots of people have been trying and not succeeding, but that's something that we're really sort of excited about trying to achieve, is trying to load one of these beta amino acids, which is a bit like the purple Lego brick, which is stretched out, right? Because the shape is quite significantly different to a normal amino acid. So I think that's the one that I would say at this point in time. But, I think also Kevlar was something we were sort of, mind mapping about the other day in the group, and that would be also pretty cool as well. But that's much further on down the line, I would say.
You tell it to make back the million it costs to do the...
Yeah, well, exactly. Yeah.
and that relates to a question about sort of which of the ARSs, tRNAs, ribosomes are the hardest to modify. You've got a whole set of different ones. Is there natural unorthogonality, a word that doesn't get used often enough, the reason why there are only 20 amino acids from 64 combinations?
Yes. So I'm not so sure about the first question. I'm not entirely sure which of the existing ones are particularly hard. It's not something I've personally worked on too much, but I do think you are right. And I think in short, yes, I think one of the reasons why having 64 codes for 20 amino acids is wise is because you can account for some of these potential mistakes that you can make during the unorthogonality of these tRNA synthetases. It's actually more to do with how the tRNA reads the code as opposed to the orthogonality. But I think in general, the orthogonality between the existing amino acids and their tRNAs and their synthetases are particularly good. But there is wobbling between the anticodon and the codon. This is getting technical, sorry. So there's wobbling between the decoding of the genetic code, which means that it's sometimes helpful to have all of, so for example, valine is encoded by, the first two are exactly the same, but the last four can be anything. So there's that subtlety, which is also useful, yeah.
Yeah, I mean, I do love the fact that wobble is literally the technical term.
That is, yeah.
It's a wonderful technical term. While we're talking about bases and matching, Barney Walker has a proper DNA question. Would it be easier to get extra codons by adding a new unnatural base pair?
That is indeed something that has been suggested. It's not something our group is looking into, but there's definitely research going on to that. I don't know too much about how that's going, but it's absolutely true. You could make new codons by making unnatural base pairs. You could also make new codes by using a quadruplet code. So instead of using just 3 letters in a row, you might be able to use 4 letters in a row, for example. That's another way to solve that problem. And that's how to get beyond 64 new amino acids. Potentially we could, instead of trying to recode the ones that exist, keep the current 64 the same and use a completely different code. So something with 4 letters, or as Barney suggested, with different base pairing characteristics. These are things that have been thought of, but there's not as much progress towards these two things yet. But yeah, they're sort of from a chemical point of view, very challenging, but very exciting ideas.
So there's still a lot of questions coming in, some more technical, but I want to step away from the really technical question to one from one of our regular listeners, anonymous attendee. Anonymous attendee says, Kim, I can see you're very passionate about what you've talked about in this talk. I find your passion very inspiring. Where does your passion come from?
It's a good question, I guess. I think it's, I think about what I want to say. I think my answer to that is I genuinely just think all of this is very, very interesting. And yeah, I think it's just, I enjoy talking about it. I enjoy learning about it. I enjoy researching. It's just genuinely quite an interesting thing, I think, for me that I'm working on. Obviously, there's a bit of survivorship bias towards the thing that you end up working on, as it were. But yeah, I think, you know, the process for me started with chemistry. And I think I was just constantly impressed by the chemistry that biology and cells was able to produce. And the efficiency with which cells are able to do chemistry, I think, is something that's always been really interesting to me. And I think that's why I've made this sort of progression from studying chemistry when I was an undergraduate here and slowly moving my way through chemical biology and now into molecular biology. So yeah, I guess that's where I would say my interest in this comes from. So yeah.
Can I just explore myself, you know, how much is it about the excitement and the enthusiasm of discovery, and how much of it is the uses? Is it
Yeah.
Which one really gets you going more?
Great question. I think this is just an important point about academic research in general. And I think for me, it's the enthusiasm. I think the former of what you said. I think it's, you know, it's not that I don't believe in the potential that the thing has, but it's true that none of this is yet demonstrated. If in some ways I'm trying to sell all of you guys a dream or, you know, a future prospect, right? But I do think academia should be working on things which, you know, people have enthusiasm for, have genuine interest in. And, you know, this is a way of developing, in my opinion, people's thought processes. And that seems like a really good thing for academia to be, you know, pursuing. So I would say, yeah, I find the enthusiasm for getting these things to work a little bit more than the utility at this point in time. But maybe that will change.
And if you get Kevlar coming out at the end, then
oh yeah, exactly.
Nice byproduct. And there's a question from Kit Masala, just exploring how much you can do with evolution. So when you're making any of these, can you evolve enzymes to make products in a cell-free extract continue? You know, can you basically use this as a new tool for evolution? So
yeah, there's quite a lot to sort of think about, I guess. The first thing is you can do unbelievable things with artificial evolution. And this was awarded the Nobel Prize in 2020 for, oh, her name's just gone out of my mind. Frances Arnold, who's, I think, one of my scientific heroes at this point in time. But you can do an unbelievable amount with artificial evolution. You can do it outside living cells. You can do it mediated by living cells as well. But I think one of your suggestions was, I think it's disappeared, but I think one of the suggestions you had was, yes, you can provide the end product and try and get the enzyme to sort of evolve towards it. That is indeed a good strategy. The reality is all of these things sort of need to be tested and a little bit and need to be tried, because often when you design these experiments, you accidentally evolve something else. That's often the case with these kinds of things. And there are lots and lots of subtleties in how you design these experiments. But it is very true that you could do this within cell-free systems, as long as you have enough, you have a good way of linking the what we call the phenotype, which is the observed outcome, with the genetic code that leads to that outcome. And there are ways you can do this in sort of in microdroplets. So if you encase microdroplets with DNA barcodes, you can do sort of that kind, you can sort of envisage experiments where you can scan through libraries of different phenotypes to try and evolve out a protein in that way without a cell. But yeah, it's a complicated question. But
can you get these sort of unnatural amino acids by mistake in a cell? Would that have similar effects? And can I also just throw in, because while we always talk about the 20 amino acids, it's sort of 20 and a bit in reality.
Yes, yeah, there is a bit, yeah. Yeah, okay. So unnatural amino acids produced by a mistake in the cell. So unlike, so there's always, often there will be some small baseline of mistakes going on in the cell. When you're copying DNA, when you're producing proteins, when you're processing amino acids, there will it's almost guaranteed that will be inevitably a mistake, right? But they happen at such a low rate that nothing is noticeable, right? In the case of accidentally incorporating one of these broken amino acids, most likely that protein will just break down and not do anything and then just be degraded. Okay, so a lot of the time you would produce more than one copy, and if you only have a small amount of the wrong amino acid, the likelihood is your population of protein molecules is largely going to be good, but there might be one or two which are wrong, and those ones can just go and be degraded by the cell, and that's okay. So a functioning cell will happily degrade these things properly and not let them persist, right? This is actually a problem in lots of disease where the proteins which don't do what they're meant to do, they don't degrade properly. That's actually a big problem in diseases like Alzheimer's, for example. The other thing, yes, the 20 and a bit. So I didn't talk about an amino acid called selenocysteine, which is a sort of, if you like, an expanded unnatural amino acid. It doesn't appear very often, but it's actually more like a post-translational modification, I would say. So what do I mean by that? It's what happens to one of the amino acids is sometimes when it's loaded to its tRNA, which is that cross structure, it can undergo a transformation. There are some proteins in some walks of life, in some domains of life, which will modify that amino acid and change its identity into something slightly different and change one of its atoms to selenium, which is quite an interesting thing. I forget exactly where this happens, but that is something that has been observed in nature. But the code doesn't change in that case. It's hard to know exactly when it's going to happen, I think.
Raise the question whether you could attack that in a similar way. But I want to finish with, I've probably got time for a couple more questions. And I want to put one from, again, anonymous attendee. Thanks for this amazing talk, Kim. It seems like we're at a really exciting time in science. In computing, we have to change how we think with the advent of quantum computers. In cosmology, there's research on hairy black holes. And here it looks as though we're on a cusp on a breakthrough in genome technology. How far do you think we can go in synthetic genomes within this decade? And how do you think this compares with the other advances we're seeing across the wide spectrum of science?
That's a great question. So let me think. So the first thing I would say is I think I myself personally, I'm really bad at predicting, I think, scientific progress. So with synthetic genomes, I can say that we're, the group that I'm working in is really keen to try and push this boat as hard as we can. We are thinking about the next stages. We're going beyond 61 and we want to start recoding further, hopefully more than just another jump of 3. So I think we're working a lot on making the process much more efficient. As I showed you before, that the process of making this was 5 people standing in a row doing the same thing for about a year and a half. It was really, really long and repetitive and arduous, doing all of the various steps. And so our first step is to try and make the process much more straightforward. Once it's more straightforward, in principle, we can test things a lot easier. And it's sort of one of these classic cases where it potentially could advance really, really quickly in a very, very short amount of time. Maybe just within the next 5 years, we'll have another synthetic genome we can play with, and we might learn new things about it. Within the next 5 years, we might have synthetic genomes or synthetic chromosomes, shall we say, in human cells. That might be something that happens in the next 5 years or so. And it's really hard to know how important those discoveries will be compared to the one that we've just had making Syn61. As for other advances across the wide spectrum of science, I think that's just too broad a question. And I wouldn't want to talk about stuff that I don't know so much about. So I might bow out of that one, but I'm sure that's a good thing to discuss over a drink at some point.
So then we'll finish with one question which you definitely know the result about. It's not outside your knowledge base. What's the most unexpected result that you've ever had from an experiment or research project?
That's a good question. I'm not sure. I can't actually think of anything that comes immediately to mind. I think I was surprised my first PhD experiment went relatively smoothly. I don't think that's a good example. Let me think. Honestly, I think the thing that's been most unexpected for me, but in hindsight, I think with hindsight, all of these things don't seem that unexpected, I guess. But I'm amongst, so this is one of my current research things, is a research interest is this genetic recoding. The other thing I'm interested in is automation of biological experiments and how to get robots to perform biological experiments. And I think sort of the general observation result, if you like, that I've observed with automation of biological experiments, I guess I'm quite surprised, or relatively surprised at I was surprised at how unwilling scientists are to actually use robots to do their experiments. So that's what I would say is the most unexpected result, I think.
Fascinating. I'm tempted to ask, is this postdocs not wanting to be replaced, or is this?
No, it's not. No, it's not. No, it's a long story, and I guess a little bit separate to this one. But it's I think it's an issue. It's an issue of time, and I think working practices, and the fact that working practices change very slowly, especially in biology. But yeah.
Absolutely. There's a magic trick which makes everything work, and you have to do it exactly the same way.
Yeah, exactly. Yeah.
Kim, thank you so much. There's a few more questions. I'm afraid we don't really have time for them all. We've already overrun slightly. So thank you very, very much for joining us. Thank you for sharing your excitement and enthusiasm about this whole area. I've certainly found it fascinating, clearly the audience has, to hear both what is happening, but also the various avenues for experimenting and to completely changing our understanding of the basic biology that many of us learned at school. So thank you very much for being with us.
Pleasure. Thank you very much for having me.